In this last video, we have seen the physiological effects, that is biopotential electrodes. We have seen about the biopotential electrodes and biopotential recording. Now in this unit we have to see about the biochemical and non-electrical parameter measurement. Non-electrical measurement. So in this regard, what are the things we are going to see? That is pH, PO2, then PCO2 measurement, calorimeter, then auto analyzer, blood flow meter, cardiac output measurement, respiratory measurement, blood pressure measurement, temperature measurement, then pulse measurement, then blood cell counters. So actually what is meant by pH measurement? In our body, the chemical balance can be determined by pH value of blood and other body fluids. That is, pH is defined as hydrogen ion concentration of a fluid. It is a logarithmic of the reciprocal of hydrogen ion concentration. So there is a pH measurement. Then what is the PO measurement? That is, PO measurement is nothing but a measurement of partial pressure of oxygen. The partial pressure of gas is proportional to the quantity of that gas present in the blood. Okay, PO2 measurement is defined as a measurement of partial pressure of oxygen. The partial pressure of proportional to the quantity of gas in the blood. Okay, the effective functioning of both respiratory and cardiovascular is can be analyzed by PO2. Then what is meant by PCO2? That is, PCO2 is nothing but a measurement of partial pressure of carbon dioxide. Okay, PCO2 is the measurement of partial carbon dioxide. PO2 is a partial pressure of oxygen. The effective functioning of both respiratory and cardiovascular system can be analyzed by PCO2 measurement. Okay, so that is the thing we have to understand regarding pH, PO2 and PCO2. Okay, let us come for the topic calorimeter. So calorimeter is nothing but a clinical test play a major role in identifying the abnormalities in human being. The blood serum is a complex fluid that contains a large number of complex fluid that contains a large number of chemical substances dissolved in it. These substances have the property of absorbing and emitting the visible light. The nature of these biological substances can be determined by analyzing their absorbance and transmittance characteristics. Mainly, protein and iron levels in blood can be measured using this calorimeter technique. So what is a blood serum? Serum is a clear yellowish colored fluid which is a part of the blood. It does not contain white or red blood cells or a clotting factor. It is the blood plasma without the fibrinogens. Okay, so let us see the types of calorimeter. Before seeing the calorimeter, we have to understand what is the principle of calorimeter. Electrophoresis is the principle of calorimeter. The electrophoresis is the movement of solid phase with respect to liquid. The buffer solution is used to carry the current and to maintain the pH value of solution as constant during the migration. Once again, repeat, electrophoresis is the movement of solid phase with respect to a liquid. The buffer solution is used to carry the current and to maintain pH value of the solution as constant during the migration. So the procedure for quantitative chemical analysis, it is mainly, that is a calorimetry is nothing but a procedure for quantitative chemical analysis. The two solutions, that is the test material and the standard solution, are observed simultaneously in a calorimeter based on comparison of color developed for visible light. Concentration of unknown solution determined on the basis of absorption of the light. So this is the principle, that is the electrophoresis. The electrophoresis is nothing but a Beer's Lambert law, that the states that concentration of solute is proportional to the absorbance. Okay, so this is the exact block diagram of a calorimeter. There is a light source, then using a lens we have to eliminate those light waves to the medium 1 and medium 2. After transmitted, the light intensity has been monitored. So how many molecules inside the liquid or a solution, that is depends upon the light intensity. That is, light intensity is depends upon those concentration of the solution. Okay, so this is the calorimeter. So calorimeter, the total absorbance of the two liquid column can be obtained by sum of individual absorbance. The amount of absorbance depends on the number of molecules present in the liquid column. So what Beer's law states? The Beer's law states that A equal to A into Cl. A equal to absorbability. C concentration of absorbing substance in cuvette. L cuvette path length. So calorimeter, the absorbability can be determined by measuring the absorption of a liquid with a known concentration, that is Cu equal to Cs Au by As. So what is As, Au, Cs, Cu? As equal to absorption of standard solution. Au absorption of unknown solution. Cs concentration of standard solution. Then Cu concentration of unknown solution. So in this graph, that is the concentration versus absorbance. The amount of absorbance depends on number of molecules present in the liquid. Okay, the amount of absorbance depends on number of molecules present in the liquid. By Beer's law, if the path length or concentration increases, the transmittance decreases and absorbance increases. This is a main point we have to note it down. If the path length or concentration increases, the transmittance decreases and absorbance increases. Okay, so this is a calorimeter or filter photometer. Always we have to say that calorimeter is always called as a filter photometer. So it is otherwise called as filter photometer. This is used for measuring the transmittance and absorbance of the solution. So this is the filter photometer. This is the filter photometer. Single beam photometer, that is it consists of a light source, lens, okay, that make a light beam parallel. Filter for wavelength. Filter, this is a filter is for wavelength selection. Covered with a sample solution, with a sample holder keeping the solution to be analyzed. Okay, the tungsten filament is a lamp. Tungsten filament lamp gives the light radiation. This light is incident on the lens. This light is incident on the lens, which make it parallel beam of light. This parallel beam of light is passed through the sample solution. After passing through a sample, the sample absorbs some light energy, transmitting the other. This transmitted light falls on the photocell. This transmitted light falls on the photocell and that generates a current. This photocurrent is indicated by the galvanometer micrometer, which is calibrated in transmittance scale. Since the photocurrent is directly proportional to the transmitted light, the transmittance scale is linear. Different scales that are calibrated for analyzing each type of constituent of a blood are used to identify the properties of blood cells. Okay, so the typically used to determine the concentration of sodium and potassium. So that is lithium Li, denotes red, potassium yellow, then K, that is a potassium violet, then sodium yellow. It is used in the analysis of blood or urine. Okay, so this is the flame photometer. Then we have to see the flame photometer. In the flame photometer, sodium is yellow, potassium is violet. That is difference between the colors and the molecules. So this is the apparatus setup of a flame photometer. Flame photometer is a device used in inorganic chemical analysis to determine the concentration of certain metal ions among the sodium, potassium, lithium, and calcium. Okay, in principle, it is a controlled flame test with the intensity of flame color quantified by, that is quantified by the photoelectric circuitry. The intensity of the color will depend on the energy that had been absorbed by the atoms that was sufficient to vaporize them. The sample is introduced to the flame at a constant rate. Filters select which colors the photometer detects and exclude. The influence of other ions before use, the device requires calibration with a series of standard solutions of the ions to be tested. So flame photometry is a crude but cheap compared to flame emission spectros. Okay, so this is the flame photometer. So then in this method, fine droplets of sample is aspired into gas flame that burns in the chimney. A known amount of lithium salt is added to the sample as a reference. As a result, red light is emitted by the lithium and yellow and violet beam are emitted due to sodium and potassium respectively. These diffracted colors are made to incident on a photodiodes. The photodetector circuits consist of a reverse biased diode in which the current flow increases as intensity of incident light increases. A calibration potentiometer is used in every channel. Since the lithium is used as a standard reference, the output of the sodium and potassium channels are calibrated in terms of differences with the known lithium. The output can be compared with the spectral illustration. Okay, so that is a flame photometer. Then followed by spectrophotometer, the general name given to the group of instruments whose principle of operation is based on the fact that a substance of clinical interest selectively absorb or emit electromagnetic energy at different wavelengths. In spectrometer, selection filter of colorimeter is replaced by a monochromator. So this is the setup of a spectrometer, spectrophotometer. So spectrophotometer can be used as a light beam intensity as a function of its color, known as spectrophotometer. Okay, the important features of a spectrophotometer is spectral bandwidth, the percentage of sample transmission, the logarithmic range of sample absorption, percentage of reflectance measurement. The spectrometer is commonly used for measurement of transmittance or reflectance of solution. However, they can also be designed to measure the diffusivity on any of the listed light ranges that usually cover around 200 nanometer to 2500 nanometer using different controls and calibrations. Within these ranges of light, calibrations are needed on the machine using standards that vary in type depending on the wavelength of the photometric determination. Okay, the advantages of double beam photometers is easier and more stable. Advantage of single beam photometer is large dynamic range, optically simpler and more compact. Okay, spectrometers use a monochromator. So this is a monochromator containing a diffraction grating to produce the analytical spectrum. The grating can either be movable or fixed. If a single detector such as photomultiplier tube or photodiode is used, the grating can be scanned stepwise so that the detector can measure the light intensity at each wavelength. Okay. Array of detectors such as charge coupled devices or photodiode arrays can be used. In such a system, the grating is fixed and the intensity of each wavelength of light is measured by different detector in the array. Different detector in the array. When making transmission measurements, spectrophotometer quantitatively compares the fraction of light that passes through a reference solution and a test solution. Then electronically compares the intensities of two signals and computes the percentage of transmission of the sample compared to the reference electrode. Okay, so this is a spectrophotometer. So in this, what are the main applications in biochemistry? So spectrophotometry is an important technique used in many biochemical experiments that involve DNA, RNA, and protein isolation, enzyme kinetics, and biochemical analysis. The spectrophotometer is used to measure colored compounds in the visible region of light, that is between 350 nanometer and 800 nanometer. Thus, it can be used to find more information about the substance being studied. In biochemical experiments, a chemical or physical property is chosen and procedure that is used in specific to the property in order to derive more information about the sample, such as quantity, purity, enzyme activity, etc. Okay, so main advantages of using a spectrophotometer is that is highly accurate instrument. That is a highly accurate instrument, very sensitive, inexpensive, relatively simple process, convenient for use in laboratories. So these are the things we have to study and learn about the spectrophotometer. Followed by auto analyzer. So this is a auto analyzer. The auto analyzer is used to measure blood chemistry and display that on a graphic reorder. In contains various blocks which are explained here. That is sample are fed into analyzer by using a sampler, mixing or proportioning pump and manifold. Okay, this is the main subsystem of a auto analyzer. Proportioning pump is basically a simple peristaltic pump. It's working simultaneously a number of tubes with a certain ratio of diameters. Then dialyzer, it separates interfacing substances from the sample by permitting selective passage of sample through a semi-permeable membrane. Next, heating bath. Heating bath, it is used to heat fluids, that is to heat fluids continuously to the exact temperature of 37 degrees Celsius. Then calorimeter, it is used to monitor the changes in optical density of the fluids which flow through the tubular flow cell. Okay, so this is all about the calorimeters. So then followed by blood flow meter. So blood flow meter, The circulatory system, that is why we are going for blood flow meter, means the circulatory system of human anatomy helps in the flow of blood throughout the body. During this process, adequate amount of blood should be supplied for the organs to perform their functions. Various diseases cause improper blood flow and supply to various parts of the body. Therefore, a disease can be diagnosed by measuring the rate of blood flow in the concerned vessel. So, the main definition of a blood flow meter measurement, that is, blood flow rate can be determined with the help of blood flow meter. The rate of flow of blood in a vessel is given as the volume of blood that passes through the vessel in a given unit of time. Okay, an adequate blood supply is necessary for all the organs of the body. Impaired supply of blood is the cause of various diseases. The ability to measure blood flow in the vessel that supplies a particular organ would be therefore of great help in diagnosing such diseases. Blood flow is a rather elusive variable that cannot be measured directly. Okay, so need for blood flow meter. So what are the needs? That is inspection for blood block in blood flow, testing artificial blood vessels during organ transplantation, during fistula creation, in dialysis. Okay, so basically it is categorized into invasive, that is surgical, and non-invasive through the skin. Okay, so what are the types of blood flow meter is magnetic blood flow meter, ultrasonic blood flow meter, thermal convection method, radiographic method, then indicator dilution method. Okay, so these are the categories of blood flow meters. So first of all, we have to see the magnetic blood flow meter, that is electromagnetic blood flow meter. It is based on the electromagnetic induction principle. When an electric conductor is moved through a magnetic field, a voltage is induced in the conductor. The voltage induced in the conductor is proportional to the velocity of motion of the conductor. So the conductive blood that flows through a tube located in the magnetic field. So this is the apparatus setup. A permanent magnet is placed. A permanent magnet is placed around the blood vessel, and it generates a magnetic field perpendicular to the direction of the blood flow. Okay, perpendicular to the direction of the blood flow. The voltage induced in the moving blood column can be measured with the help with the stationary electrodes placed on the opposite sides of the blood vessel and perpendicular to the magnetic field. So this is the general setup of a blood flow meter. Okay, so this is the block diagram. So this is the block diagram. The working of a magnetic blood flow meter can be explained with the block diagram. It consists of a electromagnet, oscillator gate, and a low pass filter. Okay, the oscillator the oscillator is used to drive the magnet. The oscillator is used to drive the magnet, and it provides control signal for the gate. The oscillator operates at a frequency between 60 Hz and 400 Hz. The gate detector makes the polarity of the output signal reverse when the flow of blood is in reverse direction. The blood flow can be easily recorded from the frequency response of the system. The average flow pulses can be obtained with the help of this low pass filter. Okay, so this is the magnetic blood flow meter, electromagnetic blood flow meter. Okay, so then ultrasonic blood flow meter. So the ultrasonic blood flow meter, so ultrasonic blood flow meter is the velocity of flowing blood is determined with the beam of ultrasonic energy. So this is the principle. Okay, there is a two type. One is transit time ultrasonic blood flow meter, another one is Doppler type. Okay, so transit time, in a transit time ultrasonic blood flow meters, a pulsed ultrasonic beam is directed at a shallow angle through the blood vessel, and the transit time is then measured. When the blood flow is in the direction of energy transmission, the transit time is short, and when the flow of blood is in the opposite direction, the transit time value is greater. Okay, then Doppler type. So Doppler type is, it consists of an oscillator operating at a frequency range of megahertz, and it excites piezoelectric transducer. The transducer is mounted to the wall. So transducer is mounted to the wall of an exposed blood vessel, and it transmits ultrasonic beam with a frequency F into the flowing blood. So here it is. We have seen from this diagram. Okay, smaller part of transmitted energy is scattered back and received by the second transducer opposite to the first one. The scattering occurs mainly due to the result of moving blood vessels. The received signal has a different frequencies due to Doppler effect. Okay, the received frequency is F plus Fd. Received frequency is F plus Fd or F minus Fd. Depending on the direction of blood flow, when the blood flow is in the same direction of the beam, the frequency is F plus Fd. When the flow is in opposite direction, this is F minus Fd. The Doppler component Fd is directly proportional to the velocity of blood flow, and the part of transmitted energy reaches the second transducer directly with the frequency being unchanged. The output is obtained with the help of frequency meter. The output is obtained with the frequency meter. The Doppler signal of the pulsating blood flow can be heard with the help of loudspeaker. Okay, it is through the loudspeaker. Then blood flow by thermal convection. When a hot object is placed in colder flowing medium, it gets cooled by thermal convection. This principle is used in this method. Here the rate of cooling is proportional to the rate of flow of medium. Okay, two methods. In one method, a thermistor in the bloodstream is kept at the constant temperature by a servo systems. The electric energy required to maintain this constant temperature is a measure of the flow rate. In another method, an electric heater is placed between two thermocouples or thermistors that are located apart along the axis of the vessel. The temperature difference between the upstream and downstream sensor is a measure of the blood velocity. This is called thermomer, thermostatmer. Okay, so another one, that is a thermal convection method. Thermal convection method is nothing but a hot object is placed in a colder flowing medium, it gets cooled by thermal convection. Okay, so that is the principle behind the thermal convection method. Okay, so then followed by radiographic method. The radiographic method nothing but the flow of blood cannot be made visible with the help of x-rays due to their same density at the surrounding tissues. Hence, to make the blood flow visible, a constant medium like a iodated organic compound is injected into the blood vessel so that the circulation pattern can be made to be visible. With the help of x-rays, the progress of a contrast medium, the obstructions and the blood flow in the vessels can be estimated. Okay, any substance can be used as an indicator if it mixes readily with blood, and its concatenation in the blood can be readily determined after mixing. The substance must be stable but should not retained by the body. It must have no toxic side effects. Okay, generally what are the indicators used in a radiographic method means an indocyanine dye, isotonic saline, and radioisotopes. Okay, these are the methods. Then indicator dilution method. In indicator dilution method, there is a two categories: open circulation method and a closed circulation method. In open circulation method, the measurement is made under the assumption that the blood is not recirculated. The indicator is injected into the blood flow continuously at the beginning time t with a constant infusion rate 1 gram per minute. A detector measures the detector measures the concentration of the downstream from the injection point. Okay, so this is a open circulation method. Then closed circulation method. In this method, the when a dye or isotope is used as an indicator, the concentration does not assume a steady state. Instead, it increases in steps whenever the recirculated indicator again passes through the detector. Okay, this method is based on assumption that the blood is being circulated. Here at first indicator is injected. Okay, here the first indicator is injected and its concentration is measured with the help of detector, and when the indicator is again recirculated, the concentration increases step by step. Okay, that is, this is the shown in the this graph. Okay, so this is the closed circulation method. Okay, there is a two types. One is a open circulation method. Here the recirculation is not considered. In closed circulation method, here the recirculation is considered. Okay, this is the main difference between the circulation methods. Okay, in here the rate of flow, that is the rate of flow always equal to 1 milligram per minute per milligrams per liter. Okay, rate of flow, that is liters per minute, how the liquid flows, that is rate of flow liters per minute equal to 1 milligram per minute divided by milligrams per liter, that is c naught. Okay, or per, it is always measured by 1 milligram per minute. Okay, then cardiac output measurement. Okay, cardiac output is the amount of blood delivered by the heart to the aorta in 1 minute. For a normal adult, the cardiac output is 4 to 6 liter per minute. The measurement of cardiac output is necessary to study the various cardiac disorders. The decrease in cardiac output is due to low pressure, reduced tissue oxygenation, poor renal function, shock, and acidosis. So these are the main symptoms due to low pressure. Okay, so the decrease in cardiac output is due to these factors. Okay, so the cardiac measures, so see was the amount of blood pumped by ventricle in 1 minute. Okay, so what are the methods in a cardiac output measure determination? Simple method, fixed method, indicator dilution method, impedance change method. Okay, these are the four methods. So simple method, in simple method we have just calculate the cardiac output equal to heart rate into stroke volume. Okay, around 5 liter, 70 beats per minute and 70 milliliter per beat. Okay, rate beats per minute, volume milliliter per minute beat. Okay, so rate is based on beats per minute, then volume milliliter per beat. Okay, so this is the simple method. Okay, we will just calculate by touching the person and calculating the beats per minute. Okay, then fixed method. Fixed principle is a straightforward application of law of mass conservation. The amount of oxygen entering lungs by venous blood plus the amount extracted from the breathed air has to be equal to the amount of oxygen leaving lungs by arterial blood. Okay, it is always said, that is in otherwise it is said by the difference between the amount of oxygen leaving lungs by arterial blood during 1 minute and the amount of entering lungs by venous blood during same time is equal the amount of O2, that is oxygen extracted from the breathed air. Okay, once again repeat, the difference between the amount of oxygen leaving lungs by arterial blood during 1 minute and the amount of blood entering lungs by venous blood during same time is equal to the amount of oxygen extracted from the breathed air. Okay, so this is the exact fixed method. So fixed method equal to I equal to CAQ minus CVQ. Okay, Q is a cardiac output in terms of liters per minute. I, that is I is nothing but a I, amount of infused or removed oxygen per unit time. Then Ca, concentration of oxygen in arterial blood, that is outgoing blood. Cv, concentration of oxygen in venous blood. Venous means incoming blood. Okay, so this is the fixed method diagram. Okay, so mixed venous blood is given, then arterial blood are measured. Okay, so outgoing blood is a Ca, that is arterial blood. Incoming blood, that is venous blood, is a CV. Okay, this is a CV. Q, cardiac output. I equal to amount of infused or removed oxygen per unit time. Okay, so this is the fixed method. Then thermal dilution method, that is indication dilution method or thermal dilution method. Using a classical method, amount of injector substance is injected into the venous system. Subsequently, time course of indicator concentration is assessed at the chosen site of the arterial tree. The distance between the application measurement site does not need to be known. The method is based on the following fact: the greater the cardiac output, the greater is the velocity of blood in the vasculature and the faster is the passage of indicator substance across the measurement site. Okay, then followed by thermodilution method. This is the thermodilution method. This is the thermodilution method. Thermodilution method, 10 percentage of 5, that is 10 ml of 5 percentage dextrose in water at room temperature is injected as a thermal indicator into the right atrium. After mixing, it is detected in the pulmonary artery by a thermistor placed at the tip of the miniature catheter probe. The difference between the indicator temperature and the circulating blood temperature in the pulmonary artery is measured. The reduction in the temperature, the reduction in the temperature is integrated with respect to time. Okay, it is displayed in output unit. After applying corrections, the cardiac output is displayed a liter per minute. Amplifier block is used to remove non-linearity of the thermistors. Okay, so what are the limitations of thermal dilution method? Affected by the phase of respiration and should be measured at the same time of respiratory cycle. Variations in the speed of cold water injection can result in altered measurement. Okay, then Impedance lithosmography or impedance change method. Electrical resistance of a chest can be measured using only few electrodes. The resistance changes during the cardiac cycle due to the changes of volume of blood inside the heart. These changes can be used for calculation of stroke volume and consequently cardiac output. The method is non-expensive and non-invasive but not accurate. Okay, so this is the impedance lithosmography or impedance change method. Okay, then what are the factors affecting the cardiac output? Heart rate, autonomic intervention, hormones, that is majorly which consider the hormones epinephrine, norepinephrine, thyroid hormone. Then cardiac reflexes, stroke volume, Starling's law, Venus return, cardiac reflexes. Okay. Then intrinsic, what are the intrinsic conditions? That is result from normal functioning characteristics of heart, that is contractility, HR, preload sketch. Extrinsic, that is involves neural and hormonal control, then automatic nervous system. Okay, so these are the factors affecting the cardiac output. Okay, so then we have to study about the respiratory rate measurement. Then in this topic, respiratory rate measurement. Okay, so rate measurement, that is mainly used for functioning of a lungs. Okay, so respiration is the interchange of gases between organism and the living medium. Respiratory system provides a means of occurring oxygen and eliminating CO2. So what are the types of respiration? Internal respiration, external respiration. Internal respiration is the exchange of gases between the bloodstream and nearby cells. External respiration is the exchange of gases between the lungs and the bloodstream. Okay, so respiration parameters. So tidal volume, increased reserve volume, expiratory reserve volume, residual volume, minute volume, total lung capacity, vital capacity, inspiratory capacity, functional residual capacity. Those are the main parameters that is used in the respiratory measurements. Okay, so these are the some of the parameters, that is inspiratory reserve volume, it is equal to 3600 ml. Then expiratory reserve volume, that is equal to 1200 ml. Residual volume, it is the amount of gas remaining in the lungs at the end of maximal expiration, it is equal to 1200 ml. So like that, vital capacity, it is the maximum amount of gas that can be expelled from the lungs by forceful effort from maximal inspiration, that is 4800 ml for a normal person. Inspiratory capacity, it is the maximum amount of gas that can be inspired from the resting expiratory level. It is the sum of tidal volume and inspiratory reserve volume, it is equal to 3600 ml for a normal person. Then total lung capacity, the total lung capacity is nothing but the amount of gas contained in the lungs at the end of the maximal inspiration. Okay, so it is a some inspiration capacity and a functional residual capacity. Its value, its level is 6000 ml. Okay, so functional residual capacity, FRC equal to ERV plus RV, that is residual volume by expiration rate volume. Okay, so this is the expiration, the respiration parameters. So what is the tidal volume, expiratory reserve volume, residual volume? This is the graph which indicates that indicated standard ml, that is 2600 ml, 3600 ml, 4000 and 5200 ml is the total volume. Based on this, the respiration rate of lungs can be measured. Okay, so these are the static lung volume and capacities we have already discussed now. Okay, so these are the general graph for the respiration measurements. Okay, for values or for young healthy male, values for females are somewhat lower. Okay, when compared with the females, here the young healthy male has more respiration rate. Okay, so respiration parameters, that is airway resistance, lung compliance, lung elasticity, intra thoracic pressure, it is the positive and negative pressure occur within the thoracic cavity. Okay, these are also the respiration parameters, that is lung volumes and capacities. Okay, so what are the types of respiration rate measurement? That is displacement measurement, thermistor, impedance pneumonography, CO2 method, apnea detectors. Okay, these are the types of respiration rate measurement. Okay, so displacement measurement, thermistor method, impedance pneumography, CO2 method, apno detectors. Okay, In the displacement method, in this method, the transducer is held by elastic band which goes around the chest. The respiratory movement results in a corresponding resistance change of the strain gauge. It is connected as one arm of the Wheatstone bridge circuit. It varies with the chest expansion. Okay, so this is the thermistor method. Generally, there is a temperature difference between inspired and expired air. This temperature is sensed by placing thermistor in front of nozzles. Thermistor is usually fitted inside the nose and generally used for newborn child. Okay, this is a thermistor method. Okay, so thermistor method, that is connected to the bridge circuit, can be used in air temperature, that is minus 60 degree to 150 degree Celsius. Then impedance pneumography. This is the indirect method of measurement. Impedance pneumography is based on the fact that the AC impedance across the chest of a patient changes as respiration occurs. That is 50 to 50 kilohertz. AC signal is produced by oscillator circuit and is given to the chest of the patient. Okay, so this is the impedance pneumography. Okay, i.e. current or chest impedance without respiration, that is, or change of del or is equal to change of chest impedance due to respiration. Okay, this is the experimental setup of impedance pneumography. Okay, then CO2 method. Respiration rate can be measured by measuring CO2. That is, CO2 measurement based on the absorption property of infrared rays by certain gases. When infrared rays are passed through the expired air, which contains certain amount of CO2, some of the radiations are absorbed by it. So there is loss of heat energy associated with the rays. The detector changes the loss in heating effect of the rays into an electrical signal. It is used to get the average respiration rate. The beam from another infrared source falls on the reference covered side. Okay, so this is the CO2 method. So gas in reference side is heated more than that on the test side. A diaphragm is pushed slightly to the test side of the detector. This diaphragm forms one plate of a capacitor. Okay, the amplified circuit is integrated. It is used for continuous monitoring the respiration rate. This is the CO2. This is the indirect method of respiratory measurement. Apnea detectors. Apnea detectors is stopping of breathing. Okay, it leads to arrest of the circulation. It can be occur at the conditions like head injury, drug overdose, etc. It can also occur in premature babies during the first week of life because of their immature nervous system. Apnea monitor is used to watch the apnea patient's respiration rate. So this is the apnea detectors. It is mainly used in ICU wards and for coma patients. Okay, so this is the apnea detectors. Here the negative detector, positive detector, comparator, then alarm, that is schemic trigger and the speaker, loudspeaker has been used for emergency purpose. Okay, the output of amplifier is connected with the motion and respiration channel blocks. The frequency below 1.5 hertz is identified as respiration, and above 1.5 is analyzed or identified as a motion. Okay, high frequency signal above threshold is sensed by positive detector. If there is no respiration, then ischemic trigger circuit gives the output to switch on the alarm. If the respiration said low pass filter is used to remove high frequency signal. Output of the alarm is connected with the speaker. So where there is no respiration for a period of 10 or 20 seconds, then audio signal through the speaker and visual signal through the flashlight is delivered. Okay, so this is the method of respiration determination. Okay, so finally we have seen the apno detectors. Okay, so apno detectors, we have seen the respiration measurement through these apno detectors. Okay, okay, so up to this we have seen the respiratory cardiac output measurements, then respiratory measurements, then blood flow meters, and pH, that is a chemical biochemical substances like pH, PO2, PCO2 has been measured during some techniques. Okay, then we have to see that blood pressure measurements, temperature measurements, and so many things. We will continue those topics in our next video. Thank you.